Now we talk about acid-base properties of amino acids. Last time in the video uploaded just now, we had talked about classification scheme and in general about amino acid we had concerned. Now in this class we come to a very specific thing, that is acid-base properties of amino acids. For that matter, the substances which can act as acid and base two in one. They are called amphoteric substances, and amino acids are the ones. They can act both as acid and as a base. What do we mean by acid and what do we mean by base? In organic chemistry में और definitions थी, but in biochemistry we say that acid is something which is a proton donor, which donates a proton. For example, ये Ha, it's a proton donor. It is donating a proton, so this is called acid. And what is a base? Base is proton acceptor. Now it is dissociating into H plus and A minus. Let me write here. Now it's a reversible reaction one proposing. So A minus will accept a proton to give rise to Ha. So proton acceptor is base, proton donor is acid. And amino acids are able to act as both. Here we can see this is the neutral form of amino acid at the physiological pH. Most, if not all, amino acid exist in this form. In the last lecture, we had seen that all amino acids have got a carboxyl group and a NH2 group. So this can be rewritten as this H यहाँ आ गया, and this becomes zwitter ion form, and most if not all amino acid exist in this form, the neutral form called zwitter ion. When the pH is decreased, then predominance of H plus will be there, and ये H यहाँ आ गया, so this becomes cation or the positive form. Just see positive. And if you increase the pH, converse series of events, it will become anionic form or the deprotonated form, you can see. We are mentioning this term Pi. This is called isoelectric pH. So when the pH equals isoelectric pH, the amino acid exists as zwitter ion. In other words, an amino acid exists as zwitter ion only when pH is equal to Pi, isoelectric pH, and isoelectric pH varies from one amino acid to another, as you can see in this chart. And when the pH falls below isoelectric pH, as you can see here, then the cationic form. When the pH is above isoelectric pH, then the anionic form. So this summarizes all these things. When this exists as cationic form or the diprotonated form, and when it exists as anionic form, when pH is less than Pi. So acid-base properties, I emphasize that now. Why zwitter ion it is called? अब अगर आप देखो, zwitter ion is capable of donating a proton. So here it is donating, it's a proton donor. So here it is an acid. Zwitter ion is also capable of accepting a proton. So here it is acting as a base. So amphoteric nature, capable of acting both as an acid and a base. We study these acid-base properties in greater details than the other properties because of certain reasons. Buffers. So this is what I have said already. So predominant form of an amino acid at its isoelectric pH is zwitter ion. Now we talk about something called buffers. Buffers are systems which resist change in the pH when a small amount of acid and base are added to it. And a buffer का composition है when a proton donor Ha and its conjugate base E minus, I write it here, when both are present in equimolar concentration, then the system acts as a buffer. So acid and conjugate base in equimolar concentration, they constitute a buffer system. So HAA minus, अब हम एमाइनो एसिड्स को देखते हैं. अब ये देखो एमाइनो एसिड at a low pH, as we had seen, it exists in the cationic state. When you start increasing the pH, dissociation of carboxylate group will occur and gradually it will reach the zwitter ion state. So now it is NH3 plus. This is the zwitter ion form. So at a lower pH, as we had seen, below isoelectric pH, it was in the cationic form. As you increase the pH, it changes to zwitter ion form. And further increase the pH, it will be converted to the anionic form. And as we have seen in the anionic form, it is COO minus, and here it is NH2, like that. So यहाँ पर there is a proton donation, dissociation of the carboxylate group. यहाँ पर there is a proton donation, dissociation of the amino group. So if you see the cationic form and the zwitter ion form together, this is cationic form is acting as an acid and zwitter ion is acting as a conjugate base. So जब titration शुरू हुआ था, it was only cationic. खत्म हुआ, it is only zwitter ion. Midway through this, cationic form की concentration zwitter ion के बराबर होगी. At this place, it will be acting as a buffer. I will show it more explicitly. First stage titration corresponding to dissociation of the carboxylate group. Midway through the titration, the concentration of cationic form is equal to concentration of the zwitter ion form, and it is acting as a buffer. Likewise, second phase corresponds to dissociation of amino group. Midway through this is zwitter ion concentration is equal to anionic form. So this is acting as a buffer. So each amino acid has got two buffering zones. The first one corresponds to dissociation of carboxylate group. Second one to dissociation of the amino group. In a diagrammatic form, I have explained everything so that there is confusion now. When we started the titration, cationic form. धीरे धीरे क्या हुआ, dissociation of the carboxylate group हुआ. यहाँ पर at the end of first phase, it is zwitter ion. So midway through this, when the cationic form की concentration was equal to zwitter ion, it is acting as a buffer. Buffer का मतलब देखिए flat zone है. ये titration curve कैसे होता है? We monitor the pH along y-axis and addition of alkali in the x-axis. So midway through the titration, you see hardly there is any rise in the pH with addition of NaOH. So this is the flat zone, is the buffering zone. इसी तरह से second flat zone is the buffering zone. तो यहाँ पर क्या होगा, इस zone में concentration of zwitter ion is nearly equal to concentration of the anionic form. All right. So one more point, what is this pK1 and pK2? इसके लिए हमें समझनी होगी Henderson-Hasselbalch equation. pH is equal to pK plus log of base concentration A minus divided by acid concentration. अब फर्स्ट फेज के अंदर एसिड क्या था? cation was acid, the proton donor. Zwitter ion was base. So midway इन दोनों की कॉन्सेंट्रेशन इक्वल थी, एसिड और बेस की. तो यहां पर इस इक्वेशन में आप देखिए, इफ द कॉन्सेंट्रेशन आर इक्वल, this ratio becomes equal to 1. And what is value of log 1? It is 0. So the equation becomes now pH is equal to pK plus log of 1, that is 0. In other words, pH becomes equal to pK. So buffering capacity is maximum when two requirements are met. One, acid की concentration base की concentration के बराबर हो, और दूसरी when pH is equal to pK. तो अब आप देख लीजिए, this is first phase corresponds to dissociation of the carboxylate group, and pK of the carboxylate group is 2.34. So every amino acid is able to act as a buffer at or around pH of 2.34. Second phase, every amino acid is able to act as buffer at or around pH of 9.69. But our body pH is 7.4 plus minus. It becomes 7.35 to 7.45. So amino acids are not able to act as buffer at the physiological pH. They are able to act as buffer at the pH of this 2.34 and 9.69, not at the body pH. So an important Viva question, I have taken quite some time to explain it, why every amino acid is not able to act as a buffer at the body pH, but there is one exception, histidine. Histidine, it has got an R group, which is called imidazole R group. It is a dissociable group. And pK of the dissociable imidazole group is somewhere near 7.4. Actually, it is 6.8 something, but it is quite near to physiological pH. So histidine K, instead of 2, it will have 3 buffering zones. Instead of 2, it will have 3 buffering zones. The first one corresponding to dissociation of carboxylate, second one to imidazole, and third one to amino group. So second one per it will be able to act as a buffer. Right. So the key points, very short point which I have taken quite some time to explain. Amino acids are zwitter ion at its isoelectric pH, no net charge. When you decrease the pH, then the cationic form. When you increase the pH above pi, it is anionic form. There are 2 buffering zones, one corresponding to dissociation of carboxylate group, other to dissociation of amino group. And why only, and that actually shows why they are not able to act as a buffer, because iska carboxylate group ka pK is 2 point something, iska 9 point something, which are far removed from body pH of 7.4. But pK value of imidazole group of histidine is relatively closer to 7.4. Hence, histidine has got capacity to act as a buffer. So we talked about amphoteric. There are 2 more terms, amphibolic, amphipathic. Something amphibian. दोनों तरफ का काम कर सकता है. So please, that's an STL for you. try to answer it.